Greetings, Life Sciences, grade 12. This is the last communication, last uh, session with you with regards to 2025. I just wanna let you go to festive season in style and with less stress. For those who just wanna make sure that how was the exam. This is the opportunity we are just going to mark now. Let's mark at Metric Hub. Let's mark ensuring academic excellence for all. I'm sure now you can confidently refer, you can confidently bring your sisters, your brothers, siblings, and family that Metric Hub is home. We have done quite a lot. It has been a good year. We really worked so together. We really worked so good together then. So, without wasting any time, I just want us go through the paper. We want to mark. We want to ensure that the paper is extensively reviewed. We are reviewing for those who were doing grade 12, coming to grade 12 now. You are welcome. Let's do it now and never doubt yourself. You are at the right place. And I really assure you that all is going to be well. So without wasting any time, we are going to go straight to our paper and then we want to work out this wallpaper right now and see what's happening with the entire session. So I'm not gonna read the instructions. These instructions you are quite aware and you are actually veterans in them. We go question one, which is multiple choice. It says a gene is dash. A gene is dashing. So if you can see, this is also a typical question that I once answered previously in the last session of preparation for these exams. A gene is a segment on a chromosome coding for a characteristic. I'm sure that you understand that it can be, we can agree that the answer is what is B. It's a segment on a chromosome coding for a characteristic. It is actually defined as a segment of DNA located on a chromosome that carries the code for specific protein or a functional RNA molecule, which in turn determines the particular characteristic or a trait. So therefore, our correct answer is what is B. I'm sure the issue of haploid number of cells, it's half the number of chromosomes that is found from one or a one set of chromosomes from either the maternal or paternal side, and then a complete set of chromosomes of an organism is something else which we can say that we can refer it to say complete set of chromosomes referring to the structure and what which we can say karyotype. But our answer is B. The significance of DNA replication is that cells formed at the end of mitosis will dash. The significance it is that DNA replication, it actually it is the formation of a DNA molecule which is genetically or which is identical to the original molecule. So the answer that is actually important here, or which is the significance, is that at the end of mitosis, we have got genetically identical cells. Genetically identical cells are formed, which is a genetically identical uh, cells are formed. I'm sure remember that DNA replication it occurs during the S phase of interphase before mitosis begins. So what you need to understand is that it allows identical cells to be formed. Which one of the following statements is a feature of punctuated equilibrium? Is a feature of punctuated equilibrium. I want you to understand is it is that there are long periods of little or no change in a population Punctuated equilibrium is it actually has long periods of little or no change in a population. What you need to understand is that it's a theory proposing that evolution occurs in rapid bursts, but speciation events followed by long periods of stability or stasis with little or no change in species. So what I want you to understand is that this contrasts with the idea of slow continuous evolutionary changes, which is gradualism. So punctuated equilibrium, it is defined with letter what? With letter C. It is defined with letter C. And then we have got homophilia, number 4. Homophilia. Homophilia is dash. Homophilia is it is one of the sex-linked diseases that I want you to take note of, and I'm sure that you remember that homophilia, we talk about color blindness and so forth, which are sex-linked diseases that I'm sure quite a lot of times we have discussed, and some of you have actually made it. I mean, it has been simplified for you there. So it is caused by mutation of an allele on an X chromosome. The disease causing allele is found on an X chromosome, which is what, which is B. It is actually caused by a mutation of allele on an X chromosome. And then non-disjunction of chromosomes in meiosis, actually it actually causes what, Down syndrome. That is one thing that we can talk about in regard to that. So the answer is B. Which one of the following is an example of heterozygous genotype? It's an example of heterozygous genotype. Also on Metric Hub channel, I have already I explained quite a lot. This is heterozygous. This is homozygous. This is homozygous because we have got recessive R. We have got homozygous recessive R. Recessive R here, and then this is also homozygous dominant. This one is also called homozygous dominant B, and then we have got also this one, which we call it is what, heterozygous, which we can say is red white, which we can say that this is heterozygous because we have got two contrasting alleles that are joined together, and then this one we can otherwise tell it again to say it is what, it's homozygous recessive. So our answer in this case is that it is what, heterozygous is C, which you can see that C is what, is heterozygous. A heterozygous, we have got a contrast or contrasting alleles that are joined together. So what you need to understand is that these combinations, they are actually leading us to understand monohybrid crosses, which we can talk about complete and incomplete dominance. So we can go to question 6. We have got a diagram representing DNA replication that is given here. A diagram that represents DNA replication. We are given here. As you can see, we have got the DNA structure that is unwinded, and we are given all those. So let us just go straight. I'm sure, remember, if we take this part alone, we say that DNA is built up or is made of nucleotides. So if we can take one piece of this, we can say this is a what? A nucleotide, which is made up of phosphate, deoxyribose, and nitrogen base. So three of them, they actually form, or they join together as a chain to form what? To form a DNA structure, which I want us to look at the questions given there. Which one of the following combinations correctly identifies molecule 1, 4, and 5? Molecule 1, we need just to see what is molecule 1. Molecule 1, molecule 1, we want molecule 1, we want 4, it's molecule 1, 4, and 5. Molecule 1, 4, and 5, and 5. So what happens here is that you are just given the structure of the nucleotide, which you are actually supposed to, which you are just supposed to understand that when given this, the answer which you need to understand is that a nucleotide is the basic monomer of DNA structure, which consists of phosphate group, deoxyribose sugar, and nitrogen base. So if you can see there in this diagram, we can see that one, it points to a one. If you look at one, it actually points in the one diagram. The ring structure with ATC. It is actually the structure of ATCG, and then we can see that which we can say nitrogen basis. We can say nitrogen basis, or we can talk about the nucleotide as a whole, and then we see that 4 it points to the sugar molecule, which is the pentagon shape, and then 5 it points to the phosphate group, the cycle with a P inside. The cycle with a P inside, it represents what? The phosphate group. So therefore, when we look at this, we can see that it is D, which we say D is net 1 is nitrogen base, 4 is the sugar, and then 5 is the phosphate, which we can see that the answer is what? Is D. It represents D is the right answer. And then which of the following combination is correct for molecule 2 and 3? For molecule 2 and 3, if you look at molecule 2 and 3, which one of the following is what is correct? Molecule 2 and 3. Molecule 2 and 3. So if we look at them, what we need to understand is that we are given these structures here. So if we can otherwise relate to it, we can see molecule 2 is paired with a molecule 2. It can be paired with a guanine. It can be paired with a guanine. The complementary base for guanine is cytosine. The complementary for 2 is what? Is cytosine. And then we can also see that 3 is paired with what? 3 is paired with it is paired with adenine. 3 is paired with adenine. So which we can say that adenine, complementary base for adenine is thymine. Therefore, we can say that the correct combination of this can be what? We can therefore say that it is 2 is thymine, and 3 is what? Is guanine. It's a matter of the base pairing. I'm sure I remember we said A, it pairs with T, and then C, it pairs with the G. This is the basic complementary pairing of bases that we have been discussing ever since in terms of what? In terms of DNA. So that is what you need basically to understand in terms of the pairing, the complementary pairing of bases. In a certain plant species, one gene controls stem color and another controls plant height. The stem color can be brown or red, while the plant height can be tall or short. So what need to be understood is that we are given stem color and plant height, plant 1 and plant 2. the stem color is brown and the height is short. The plant color, the plant 2 stem is red and height is tall. So when plant 1 was crossed with plant 2, some of their offsprings were red. When 1 was crossed, we say 1 it is brown and what and short was crossed with when it was crossed with 2. So we are saying brown short is crossed with it is crossed with red toe. It is crossed with a red toe. So what happens is that when this crossed, where some of the offsprings were red and short, it resulted in what? In red and what? And short. It resulted in red and short offsprings. So now, what we need to understand is that given this, so yeah, class, therefore now the question is saying, we have got we have got plant 1 was crossed with plant 2. Some of the offsprings were red and others were short. So we are saying which one of the following represent the genotype of plant 1 and plant 2. So if we look at this expression, I want you to understand this. We are looking at a dihybrid cross here. We are looking at a dihybrid cross. So one thing that happens there is that we have got 2 characteristics that are being worked at or which are considered at the same time. So what we need to understand is that uh, in order for us to say that the problem states that some offsprings were red and others were short, we are saying offsprings, we have got offsprings, we have got some red and then we have some short. So in this regard, what you need to understand is that red, both of these characteristics are what? These are characteristics which are all recessive alleles. So for red, we are saying that these are all, for them to be red, we said red is recessive, and for short, it is also recessive. We should have homozygous T recessive like that. So to produce offsprings with both recessive alleles, in order for us to have to have offsprings with both alleles that are recessive that are recessive B, what happens is that both parents must contribute a recessive allele from what? From either of the parents. So for us to have red, it means both parents contributed a recessive B, and then which we need to know that plant 1 is brown dominant. So its genotype must be what? It is brown dominant. Hence, we are saying for it to be brown dominant, it means that its genotype, the genotype of this plant, therefore it must be what? The genotype of this plant must be heterozygous B. So this, in terms of the brown it must be brown and recessive, because we say that it has to have a dominant brown. So red is recessive, as we have seen there. And then plant 2 is red, the plant so we are saying plant 2 is red recessive, so its genotype will be what? Its genotype for that is gonna be BB. To produce an offspring that is giving us a short. For us to produce an offspring that gives us short, it means both parents must contribute a recessive T also. Both parents must have a recessive T for us to have a short plant, or to have a short allele there. So plant 1 is short recessive, so its genotype is also going to be what? Is going to be TT. Its genotype should also be TT. So plant 2 is tall dominant, so its genotype is what? The genotype of the plant 2 height, we are saying plant 2 height it is supposed to be what is going to be heterozygous T. Is going to be heterozygous T because we are saying that it is T dominant. So its genotype must be T. It must be heterozygous T there. So therefore it carries a recessive T allele. So if we look at this, what happens is that this can actually therefore tell us that the correct answer can give us to say we have got uh, we have got red toe and brown short that are crossed together. So therefore we can have some of the offsprings that are shorty and what that are shorty and others which are also red, because we are saying that the red must come from what from both parents contributing a recessive allele, and also in terms of the shortness it must also both parents contributing the recessive allele. Therefore what we are saying is that we should have D as the correct answer to say that we have got BT and homozygous PT heterozygous as expressed in the answer D there. And then the number of possible different genotypes expected in the offspring. The number of possible different genotypes expected in the offspring from this possible genotypes that are expected in the offspring of this cross. I'm sure you remember that when you talk about a dihybrid cross, the dihybrid cross, when we actually work on it, it should give you to say that we can actually expect uh, the total genotypes that can be expected there. We can have we said it is uh it's gonna give us 80, which we can say that it is because we have got BBT with BT for the same gene, for the first gene stem color. It is gonna give us uh, heterozygous B crossed with recessive B there, uh, and then you'll see that we can have uh, total number of possible genotype is the product of the possibilities for each independent gene that must come there. So we have got 2 times 2, which is called 4 possible. We have got 4 possible genotypes in the offspring. And then we can therefore say that this can therefore say us that we can say our answer can give us, it's gonna give us uh, B. A rare form of rickets is caused by a dominant allele on the X chromosome. It is caused by a rare form of rickets is caused by a dominant allele on the X chromosome. Which one of the following statements is true about the inheritance of this form of rickets? Which one is correct in this form of rickets? I'm sure you can see that this is a sex-linked, which is caused by a dominant allele on an X chromosome. So what happens is that uh, it is a sex-linked allele, or it is a sex-linked disease, in other words. So what happens is that if the father is affected, all the daughters will be affected. If the father is affected, all the daughters, all the daughters or his daughters will be affected also, because if the father is affected, it means the affected allele will be also going to the what? If the father is affected, all the daughters are affected. And then we want to go to terminologies and what we call biological terms. The bond formed between two amino acids during protein synthesis, which we say the peptide bond. I'm sure this is continuously peptide. It is a peptide bond. And then an organelle that contains DNA, which is used in the tracing of female, which is used in the tracing of female ancestry. We say that it is mitochondrial DNA. It is mitochondrial DNA. It is mitochondrial DNA, which we say it is mitochondrial DNA. Mitochondrial DNA, and then undifferentiated cells in animals that have the potential to become any type of tissue. Undifferentiated cells in animals that have got a potential to become any type of tissue. I remember that we've been talking about stem cell research. We say it is what? Stem cells. And then we can talk about a change in the sequence of nitrogenous bases resulting in the formation of a different protein. I'm sure I remember this. We talk about mutation. Mutation, it is a change in the sequence of nitrogen bases. It is called mutation. And then a natural shape of the DNA molecule, it is actually a double helix. We talked about the double helix structure. It is just double helix, and then which we said that the structure of DNA, it is something like this, which we say it's a double helix. It's a double helix. The strands are twisted and rolling on top of each other. So we say that is a double helix. The permanent disappearance of species from Earth, we call that extinction. When all organisms are no longer existing, we call that extinction. We say it is extinction, which is if they are still living, we say it is what? It's called extent. The opposite of extinction is extent. A type of variation with a range of intermediate phenotypes, we say that is called continuous variation. I also even explained it where I gave the graphs that are supposed to be used to explain this. There are some graphs that we also used to explain these scenarios. An allele that is expressed in a phenotype in the heterozygous condition, an allele that is expressed in a phenotype in a heterozygous condition, we say that it's called the dominant allele. It's called the dominant. It's called the dominant allele. I was giving you this in the beginning there to say we can say heterozygous. In the heterozygous condition, it is this, which we say that is equals to where dominant T is representing TO. So dominant T is given to mean that the plant is TO, whether there is even if there is a recessive allele that is homozygous, recessive T there. So it is what is a dominant allele. So this T is what you call this T. That T is the dominant, and the small t is the recessive. I'm sure you still remember these expressions. The type, the position of an allele, on a chromosome. We have been talking about also the structure of the structures of this one. We say that the position of an allele on a chromosome, we say it's called a locus. Variation of a gene, we say it's an allele. That is these terminologies. I'm sure you can be able to relate and answer them very well. And then we have to go to question 1.3, which is either A, both or none. The phase of meiosis during which non-disjunction can occur. Uh, when do we have non-disjunction occurring? It can occur in anaphase 1 or 2. So we can say it is both non-separation, both A and B. And then we have got representation of the number, shape, and arrangement of all the chromosomes in the nucleus. The number shape, we say that is karyotype. Karyotyping, it is B only. The phylogenetic tree, it's a diagram that shows evolutionary trends or evolutionary relationship. The alleles of a gene separate into the alleles of a gene separate into different gametes during meiosis. We say that this also happens. The alleles of a gene separate into separate gametes during meiosis. They do so during the law of we say that is law of segregation. Law of segregation, which is B, and then we can go to the next question. Diagrams below represent cells from the same organism during different phases of mitosis and meiosis, which is given here. So we are given these cells there. The question is saying identify. This is one also key question that I was always saying. As a class, you should always understand that these questions, they are there are quite a lot of times given where you cannot go to exam without being able to label that diagram. So identify part A. Part A, we can say identify part A, part B, and C. Part A, part B, part C. If you can look at this, we can see that A, it is the cell membrane. Part B is the spindle fibers, as you can see. B is the spindle fiber. Which we say that it is there to control the movement of chromosomes in order to effect the process of meiosis. And then C is the centriole, which we can see there. Sometimes they can also call it a pore, from which the spindle fibers are attached in order for them to move and control the processes that we are talking about. And then which diagrams VW, XYZ represents a phase, represents a cell during prophase of mitosis, which we are saying during the prophase of mitosis, you need to see that when my metaphase, I was saying that you need always to know that these these processes, you just need to know that when we look at structure and process of meiosis, it is one thing that you cannot omit whenever you do preparation for matric examinations, whether it is mid-year or whatsoever. Meiosis is always there. So process of mitosis. So we can say that it can be. We need to know that process of mitosis. It can be represented by W. Process of mitosis can be W. which we say that chromosomes are condensed but randomly arranged, beginning to form spindle fibers, and then which is actually one of the typical identities of prophase of mitosis, are usually scattered. The chromosomes are also scattered, and it actually showing, if you can see also W, it starts to show also the it starts to show also homologous pairs starting to associate, which are starting to associate, which looks more like prophase one of meiosis, but given the options, we can actually say that W is the best fit for general prophase where chromosome contains. So we can say W, and then the other one, which we say that metaphase one, homologous chromosomes pair are aligned along the metaphase or at the what? Metaphase plate or the equator, which we can say that can be seen by what? By diagram Y. That can be Y because we say that in metaphase, homologous chromosomes, they pair, homologous chromosome pairs are aligned along the metaphase plate, which we can say it is the equator. So we can say that this is metaphase one. Metaphase one, we can say that is what? That is Y. And then prophase one, it is actually prophase one, we can say that homologous chromosomes are paired in what? They are homologous chromosomes. They are actually paired up, crossing over is visible, which should happen in what? Which must happen in a structure, which we can say that we can say V can show that V can represent prophase one. V, because we are saying that homologous chromosomes are what? They have to be paired up, crossing over is actually supposed to be somehow visible, which we can see having to take place there. And then how many chromosomes will therefore be in each cell at the end of mitosis? At the end of mitosis, how many chromosomes will be present at the end of mitosis? I want you to understand that the original somatic cell has 4 chromosomes, which is 2n is equals to 4. Mitosis produces genetically identical daughter cells with the same number of chromosomes as the parent cell. So we should have that this can give us what? It must give us 4. We are saying that mitosis produces genetically identical daughter cells with the same number of chromosomes as the parent cell. And then therefore we can see that that can be 4. And then meiosis 1, as you can see also, I'm sure you remember that meiosis, it actually has to have half the number. If we have got 4, it means we should have meiosis separates homologous pairs, reducing the chromosome number by half, which means to say haploid number, which is in the original cell, has got 4 chromosomes. So if it is the original having 4, so at the end of meiosis 1, it must have half, which is haploid. Therefore, it must have we are saying haploid, which is actually given as what? As haploid, which we say that it is what? It is N. And then remember here we say that mitosis, it is 2N. This one we said it must be 2N, which is also equals to what? To 4, as we have said. The diagram represents a stage of protein synthesis. Each amino acid, P, Q, R, and S are coded for by different codes, by different codon on the mRNA molecule. So we are given this. This question is just a repetition of a previous paper that we worked as a preparatory examination of last year. We have worked it out already for those who have been following. It is a repetition. It's just the same question that we have answered severally here. So the question is saying identify the stage of protein synthesis represented by molecule 1. The stage of protein synthesis represented, uh, identify the stage of protein synthesis that is represented by stage 1. And then when you look at these, I want you to understand that when you look at these processes, you need to understand that when we have got protein, this one, it actually represents the process of protein formation, which we talk about the what, uh, the codons and the anticodons entering and mixing together in order for them to form proteins. So it is actually supposed to say the stage of protein synthesis represented by molecule 1. It is actually translation. We are given the we are given the tRNA that is given there, and organelle Z, it is the ribosome. I once answered it again that it is the ribosome. Ribosome is the organelle that is involved in what? In protein synthesis. And then state the sequence of nitrogenous bases on the DNA molecule for codon 3 and 4. The sequence of nitrogen bases for codon, we are saying for codon 3 and 4. For codon 3 and 4, if you can see codon 3, we have seen that codon 3, we are given here that we have got our GCA. We are given that the codon 3 on the mRNA has got a sequence of on the mRNA, it has got the sequence of it has got this, which we say that using the complementary base pairing rules, the DNA synthesizes A, it pairs with T, and C pairs with G. The corresponding DNA sequence is just that, to say that when given this, we can say that G pairs with what? With the C, and then we are given this G pairs with what? With the C, and then C pairs with what? With the G, and then A pairs with what? With the T. In terms of the basic pairing of the nitrogen bases. So we are given GCA. 3 is given GCA, so it can be CT. It is supposed to be CGT, according to CGT. This is basic complementary base pairing that we have been talking about. And then 4, if we have got 4 there, 4 is actually given there. So what happens is that in this question, what you need to understand is that we are saying this base pairing, we can see that 4 on the mRNA is the sequence of, we need to see the sequence of 4 there. and then we can be able to relate them and answer them properly. So if we can see, we can say A, AUC, AUC, it has to be saying that what is the corresponding uh, uh, the corresponding anticodon that comes to this. So when we look at this again, we can see that the mRNA has the sequence GG. The mRNA, it has got the sequence, it has the sequence GGC, GGC, that is the mRNA. It has got GGC. So using the complementary base pairing rules for DNA synthesis, A pairs with T and also C pairs with G. The corresponding DNA sequence is what we can say G. The corresponding base pairing here, G, it pairs with the C, and then G, it pairs with the C again. And again, we have got C, it pairs with what? With the G. So what happens is that this can give us to say the DNA sequence, which is there, is supposed to be CCG. Which amino acid is coded for by codon by which amino acid is coded for by codon 5? Which amino acid is coded for by codon 5? Codon 5, codon 5, which amino acid is coded for by codon 5? 5, it is CGA. So what we need to understand is that 5 is CGA. So if we see that we need to understand to say when we are in this again, it needs to with the amino acid that is coded for by 5. It is actually supposed to mean that it has got a sequence of CGA. 5 has got a sequence. It has got a sequence of CGA, and then we need to find actually we actually have to find which amino acid PQRS is attached to the transfer RNA with the complementary anticodon. The complementary anticodon, and then what happens is that the tRNA molecule pairing with CGA has the anticodon GCU, where G pairs with the C pairs with the G, and A pairs with what? With U. So the diagram shows the tRNA carrying amino acids. S is the anticodon GCU. So what we can say is that S has got the amino acid GCU. S has got an amino acid GCU. So when we look at that, we can say that S is coded for by what? It is S is coded for by 5. We have got question 2, represent the sex chromosomes in a female. The sex chromosomes in a female, identify structure A. Identify structure A. A, as you can see, it is the centromere. A, it is the centromere which joins two chromatids together. Is the centromere, give the biological term for the sex chromosomes. The sex chromosomes are also called gonosomes. These are also called they are called gonosomes, and then describe how chromosome would differ in the somatic cell of a male. A male is called actually a male. The somatic cells explain how these chromosomes would differ in the somatic cell of a male. So a male would have one X chromosome and one Y chromosome, whereas female has got two X chromosomes. A male is one X and also Y chromosomes. Whereas in female, there is XX, there is two X chromosomes. There is two X chromosomes that I think we are quite aware that female, many, we say for many, we have got XY, and then for female, we have got XX. That is the difference that we need to note, and that is what we should understand. An exchange of genetic material takes place at region P. Name the process. An exchange of genetic material takes place at region P. Name the process. That name the process above. If exchange of genetic material occurs, we call that what? We call that crossing over. Crossing over. Crossing over occurs. It is crossing over. Identify the phase during which the process named occur. It occurs in meiosis, which is in prophase 1. I'm sure I remember that it occurs in prophase 1. Give one reason why the process named is important. The process, why it is important. It actually it increases genetic variation. It increases genetic variation. Increases genetic variation and improves biodiversity. So actually, meiosis, it also, we are saying it increases genetic variation. Genetic variation is increased, which also increases survival of organisms. So explain how the structure of the chromosome would differ at the end of meiosis. Explain how the structure would differ at the end of meiosis. So what happens is that at the end of meiosis 2, the sister chromatids separate. Therefore, each chromosome in the resulting gamete will consist of a single chromatid, not two, shown in the diagram. Here, each chromosome each chromosome in the resulting gamete will consist of a single chromatid. Each chromosome each chromosome in the what, it will actually have uh, each chromosome in the resulting gamete actually a resulting gamete will consist of a single chromatide, consisting of a single chromatide, will consist of a single chromatide not two as shown in the diagram, not two as shown in the diagram. So what happens is then number 6, non-disjunction, we say that is called non-separation. Non-separation of chromosomes. Non-separation of chromosomes occurred during meiosis, and a gamete with an extra chromosome is formed. If the gamete is fertilized by a normal gamete, the resulting gamete will not be normal. Explain the genetic composition of these zygotes. This is what we have said before, and I've answered this question severally. The resulting gamete, what happens is that the resulting gamete will be actually be abnormal. The resulting zygote will be abnormal. Resulting zygote will be abnormal. It will be abnormal, which I said last time that will be an abnormal, will be abnormal, and we have extra number of chromosomes. Resulting in an extra number of chromosomes. It will be resulting in extra number of chromosomes. So the gamete results in an abnormal zygote count. So what happens is that it can either have one extra or one missing zygote, which we can have either to have triple X or XXY, which can cause some Turner syndrome leading to various genetic disorders, sometimes which can actually be causing Down syndrome. We are given the analysis of sample obtained at a crime scene, a sample from victim and samples from two suspects. In their investigation, police conducted comparison of DNA to identify the criminal. I'm sure for some of you have seen again this, we have done it also just towards exams. This same question was done just towards exams there. So the first question is saying, name the technique used in criminal investigation. This we call that, it's called DNA fingerprinting. DNA fingerprinting, or we can say DNA profiling. DNA fingerprinting or DNA profiling. And then which of the suspects was at the crime scene. Which of the suspect was at the crime scene, we can look at the likeliness. Sample from suspect 2, sample from suspect 1, sample from the victim, and then the sample from the crime scene. So when we can look at it, sample at the crime scene, this is the crime sample from the crime scene. We should look the one that has got as which one of the suspect was at the crime scene. We should look at the likeliness of the samples to represent to fit to the one at the crime scene. At the crime scene, we are seeing that we can look at this, which one of them is likely to be at the crime scene. We have got this one. I'm saying we look at this bar, big bar, and then we have got also this big bar. We have got a small bar. We have got a small bar there. We have got this one. We have got that one. We have this one. We have that one, that one. We have that one. We have this one. We have that one. Sample from the crime scene. We also have this. It matches with that one. We have that. It matches. This one. It matches. It matches. It matches. It matches. So we are saying which of the suspect was at the crime scene. We can see that suspect one. We can see that one was the one suspect. We can say suspect one, give a reason. We are saying that the DNA bars, they are actually matching the DNA sample of suspect one. Suspect one, it matches one, it matches the one from the crime scene. As I was marking there, you can see that these profiles, they are actually matching. We can see a typical matching of suspect one to the one at the sample from the crime scene. We are saying they are quite matching. So there's a DNA sample of suspect one. It matches that one from the crime scene. So it means that he is the one who was at the crime scene. And then give two possible reasons for why the suspect identified in question might not be convicted if only the evidence is used. Uh, what might happen is that the DNA bends, we are saying this can because the DNA might be planted, DNA sample may have been planted. It may have been planted at the crime scene. Someone might plant the DNA sample of this person at the crime scene. Therefore, it is true that the sample is correct, but it might not be his. So we can also maybe say that he might have been, he might have been at the place before the crime. Before the crime happened, it might happen that the person was there at the place, but he is not the one who committed the crime. So we can say that that might have happened that the person was at the crime scene before the thing, and then he might also have an identical twin, which can also be an identical twin, which can also cause the DNA profiles to be similar, or it can be also human error in the laboratory during the collection, handling, and analysis of the samples that may compromise the results to say that he is the one who is a suspect or uh, who has committed the crime. So possibilities of human errors might have happened in terms of the handling and collection and analysis of the samples. Uh, the evidence could be also uh, be be dismissible as a result of improper procedure or issue with the chain of custody, which can also happen in terms of people conviction, that it can be dismissed due to improper procedures that might have been given in regard or an issue with the chain of custody that has been followed in terms of the conviction of this suspect. And then 2.25, explain how the technique named in the above, but explain how the technique named is used in paternity testing. Uh, in terms of paternity testing, DNA samples are collected from the mother with the mother, the child, and the potential fathers. DNA samples, DNA samples are collected. They are collected from the mother, from the mother, from the mother, the child, and then from the mother, the child, and the potential fathers, which we can also need to understand that when that happens, we can now also uh, from the mother, the child, and potential father, which we can say it can be fathers, potential father, and then the DNA fingerprinting is performed on all samples to create unique bands of pattern. DNA fingerprinting is performed on all samples in order to obtain, in order to create unique band patterns, to create unique band patterns. So what happened is that the child inherited half of the DNA from the mother and half from the biological father. The father's the child inherits half of the DNA from the mother and half from the biological father. The child's parenting pattern is compared to the mother's and the potential father's patterns. Any bends in the child's profile that do not match the mother's profile must match the biological father's profile. That is what you need to understand there. So, if all non-maternal bends match the potential father's bends, he is confirmed as the biological father. If they do not match, he is excluded to be the father of the child. So it's a matter of comparing the bends, and we are saying, you should understand that when a child is born, the child inherits the characteristics from what? From both parents. So what happens is that they are compared against the mother and the potential father's patterns. That is what you need to understand. We have got CADASIL is an autosomal genetic disorder caused by dominant D. It results in thickening of walls of blood vessels. The diagram shows inheritance of this one in a family. It is showing the inheritance of the disease in the family. So Name the type of diagram represented above. The type of diagram, it is called the pedigree chart or pedigree diagram. This one is called a pedigree diagram. It's called pedigree diagram or a chart. You can call it a pedigree diagram or a chart, which can also sometimes they can call it a pedigree table, which shows the lines of inheritance in a family. It identifies the lines of inheritance in a family. How many offsprings do parents 3 and 4 have? How many offsprings do parents 3 and 4 have? Parents 3 and 4. Parents 3 and 4 is this one. And this one, they have one. They have only one, who is number 80. Parent 3 and 4, how many children do they, how many offsprings? They have one. They have one, which is actually represented by 8 there. I think you remember that individuals 3 and 4 are linked by horizontal lines that we need to know that they indicate they are parents. A vertical line extends down to their children. They are actually connected in below there. So what happens is that in terms of 3 and 4, they actually give number 8 as their children, which is showing an extension of the vertical line there. And then next question is saying, give the phenotype of individual 10. Give the phenotype of individual 10. The phenotype of individual 10 is actually a female with an affected female, a female with CADASIL. 10 is what? It's a male with the condition. 10, it is the female with CADASIL is affected female or a female with CADASIL condition. It's a female with CADASIL condition. And then genotype of individual 5. Genotype of individual 5. Genotype of individual 5. We are seeing that here it is an affected. This is an affected mother, and then is marrying an affected father. Both of them are affected mother and affected father. They marry each other. So what is happening is that this child, she can be actually having one allele affected, which we can say that this can be the genotype of this person. She is supposed to be it's a individual 5. We can prove that. We can try to prove that by saying this is an affected. We say that this is caused by a recessive, by a dominant allele, and then we are saying it is crossed with what? With an affected father. So if we can say that, we say this can be giving us D, and then it's going to cross with D here. and then we have got Y. So if we cross this together, we are going to see dt, and then this one and that one is gonna give us dy. It's like that. So if you can look at it like that, if we can look at it in that regard, therefore, we are saying this girl, can she be clean? Can we say that she is clean? Individual 5. I think this person, she must be a career. She must be affected. We can see that if we look at it, let's just keep on, let me calculate it again. The mother is affected and the father is affected. The father and the mother are both affected. Therefore, this person, for a mother to be affected, it means the condition is as follows, that there is homozygous D. So this is an affected, this can be an affected child here. She is affected. I can say this can be, according to the proofing of the question there, this child must be affected. This must be an affected child. This is number 5, question B. And then using evidence from the diagram, explain why individual 1 and 2 are both heterozygous. Using evidence from the graph, why can explain why individual 1 and 2 are both heterozygous. 1 and 2, 1 and 2. If we say that this, according to this, we are saying why individual 1 and 2 are both heterozygous. We are saying this can be heterozygous because what happens is that we are having unaffected children there. That is because we have got some two sons who are unaffected there. This, this question, I'm saying it can just be proven also by calculating that to say, how can we say that they are heterozygous? If we say that they are heterozygous 1 and 2, if we say individual 1 and 2 are heterozygous, it means we are going to say the mother, she is not affected. She is, she has got this condition, as to say she is a carrier. The mother is a carrier, if we can say that. And then the father is also having, according to this, but we are saying according to this, it's going to be like saying it is heterozygous. So It's a matter of calculating those. What is the percentage chance of parents 7 and 8 having another child with uh, uh, 7 and 8 percentage chance of them having this one, percentage chance of them having another child with the condition is going to be 3 over 4. Actually, they already, total children they have, total children they have is equals to, they have actually total children, they have 7 and 8, they have got 3 children, they have 3, and then number affected is equals to 2, and then percentage chance of fourth child to be affected is equals to affected already is equals to 2 plus 1 over total over total children, which is equals to 3 over 4. So we are saying the question is saying percentage. So 3 over 4, which is equals to 3 over 4 multiplied by 100 over 1. That is equals to give us 75%. 75%. The graph shows the distribution of blood groups for 2 ethnic groups, 1 and 2, in certain country during 2021. So we are given that ethnic 1 is shaded and ethnic 2 is unshaded. And then we are given that. So therefore the question is saying which blood group is most common in both ethnic groups. Most common in both groups. Which blood group is common in both groups? We are saying which blood group is common in both groups. So we can say that the most common in ethnic, when you look at the analysis, if we look in this one, we can say that we can say there is O here, there is O, which has got the highest in both, O has got the highest in both group 1 and group 2, which you say O has got 45% in what? In ethnic 1 and 51% in what? In ethnic 2. So what we are saying is the highest, the most commonest one is what? Blood group O. Blood group O is the most commonest one there. And then the next question is saying, yes, the highest percentage in which blood group has got the highest percentage, higher percentage in ethnic 1 than in ethnic 2. Higher percentage in ethnic 1 than ethnic 2. We just have to see. Higher percentage in ethnic 1, it's 46, 40, and so it is blood group A. Higher percentage in ethnic 1 than in ethnic 2. So it's blood group A. As you can see, we have got higher percentage in 40% there, and 26%, that is A. blood group A, which you see that we have got what? 40% and then 26%. I'm saying we have got 40% versus 26%, which is given there. State all the possible genotypes. State all the possible genotypes which state all the possible genotypes of 11% of the people in ethnic 1. All the possible genotype, we are saying blood group B, which you are saying all the possible genotype blood group of 11% of this one, all the possible. It's just a matter of you understanding that blood group B is represented. When you talk about blood group B, it can be B dominant, or we can have B recessive like this. It's a matter of understanding the representation of blood B, which is asked in this regard. And then we are given that ethnic group 1 had a population of 360,000 in 2021, which blood group is 30. We are saying ethnic 1 is a population of 360,000 people in 2021, which blood group A, B, C, A, B, A, B, or O, at 39,600 people. So we need to calculate the population, calculate the percentage of the population that 39 people represent. In order for us to calculate this one, we should say percentage is equals to number of people over total population, number of people over total population. Number of people over total population multiplied by 100%. So we can calculate that to say that number of people over total population. So we have got 39,600 over total population, which is 360,000 multiplied by 100 over 1. So this is going to give us 0.11 multiplied by 100, which is going to give us 11%. That is going to give us 11%. So we should identify the blood group in the ethnic group 1 with a percentage that we are given there, 11%, which is blood group what? B. 11% is represented by what? By blood group B. Is blood group B. Is blood group B, which has got this percentage, ne? Blood group B. And then we can go to the next question. He said, explain the inheritance of blood group AB. 2.44, inheritance of blood group AB. Blood group AB. the inheritance of blood group AB, it actually need to understand that this is a codominance. It is a it is a codominance where allele A and allele B are both expressed in the individual, resulting in blood group AB, resulting in blood AB. So this is a combination of A plus what? Plus B, which give us a what? AB. That is the inheritance of blood AB class. 2.5, it is polymorphosis. It is polymorphosis are bred by horse breeders for their coat color. They are bred for their coat color. A polymo has a golden colored coat. It is the offspring of a horse with a cream colored coat and a horse with a chestnut colored coat, which are given the so what happens is that name the type of dominance displayed in the inheritance of coat color in polymorphosis. We say that in terms of this, the type of dominance I once gave you again class previously that you should be able to to answer questions of dominance. So name the type of dominance displayed on coat color in polymorphosis. So a polymorphosis is a golden color coat. It is an offspring with a cream color coat A and a horse with a chestnut color coat G. So what happens is that what happens here is it is actually we can say that this can be codominance, as sometimes it is treated as such, but usually incomplete dominance for the blended phenotype can actually show this. It is an incomplete dominance. Incomplete dominance. It is an incomplete dominance. And then why do we saw neither of the coat colors is equal to neither of the alleles for cream. It's a cream coat, nor the allele for chestnut coat, nor chestnut coat is completely dominant over the other. Breeding of horses with desired characteristics is a type of biotechnology. Name this. It is a type of biotechnology. Name the type of biotechnology. It is selective breeding or artificial selection. Artificial or selective. It is artificial or selective breeding. Name the two phenotypes of the horses that must be bred for 100% chance of polymo horse. It must be cream coat color and a chestnut coat color. This must be, we must have a polymo horse. It is supposed to be a polymo horse is or has a genotype. It has a genotype of AG. So what must happen is that we must have, so in order for us to get AG, you must cross a homozygous dominant with a homozygous dominant, this one. So we must have, which is a product of AA and GG, which can give us that. So we must cross this, therefore means that we must cross AA, we must cross AA, and it is crossed with what? With the GG equals to AG. That is what must happen there. And then we are given that a horse with a cream coat color was crossed with a Palomar horse. Use a genetic cross to show the expected phenotype. We have done this quite a lot of times also. A horse with a cream coat color was crossed with a Palomar horse. So we are saying in terms of this, we say P1 phenotype. P1 phenotype, we are saying cream colored horse cream horse is crossed with Palimo, and then we say genotype. Genotype of this one, we say that a cream horse is going to be AA and is crossed with we are saying it is crossed with a Palomar horse which is AG, and then we say that meiosis and gametes, we are going to have this, we're gonna give us that one, and then we're gonna have AG, and then we say fertilization. We are going to have AA, and then we cross that and that, we have AG. So we've got 1%, we have got one cream horse as to one Palemo, which is a 50% chance. that is 50% cream as to 50% Palim. That is what you do in this case, class. It's a matter of you being able to understand these genotype phenotype codes. It's a matter of you understanding the genotype phenotype codes. So when you do this, obviously we are saying this is genotype again, and then this is the phenotype. So you've got 1:1, which is your answer that you can get the. Yeah, thank you very much for your time. I think we can see question 2. Question 3, we are just going to work it separately on its own. It's an evolution question. We are going to work it separately just to cut our presentation short. We are going to cut this. We are going to present question 3 on its own. So let's just end on question. Let's just end on question 2. And thank you very much for your time.